In commercial poultry production worldwide, the incidence of infectious bronchitis is of major economic concern. This highly contagious and widespread disease takes its name from the initial respiratory signs, but also affects the urogenital tract. It causes poor performance in broilers and reduces egg production as well as external and internal egg quality in breeders and layers. Additionally, the birds develop a heightened susceptibility to secondary infections that can lead to a higher mortality rate and an increased use of antibiotics. The disease is caused by the infectious bronchitis virus, or IBV, transmitted via the inhalation of droplets through the upper respiratory tract. IBV shows a number of characteristic spikes on its surface. These consist of proteins and play an important role in the onset of infection and the corresponding immune response. After entering the bird's body, IBV quickly reaches its primary targets, the trachea and the bronchi. The upper respiratory tract is lined with mobile hair-like structures, the cilia. Among them are found mucus-producing cells, so-called goblet cells. Together with the mucus, the cilia provide a self-clearing mechanism effective against many invading pathogens. However, in unprotected birds, IBV is able to overcome this natural barrier and binds to the cells on the surface of the trachea. In doing so, it uses its spikes to attach itself to receptors on the cell membrane. Then the virus pushes on into the interior of the cell. Here it multiplies and subsequently infects other cells, which in turn produce many more viruses. As a result of infection, the cells lose their cilia. In this way, the natural barrier against invading pathogens is progressively destroyed, opening the door for secondary infections with other viruses or pathogenic bacteria. The initial binding to the receptors on the cell membrane by means of the spikes on the surface of the IB virus is a decisive step in the onset of infection. If the binding can be prevented, infection will not occur. Timely vaccination is a way to achieve this. It results in a prompt immune response based on local mucosal immunity, which includes the local production of antibodies and their secretion into the mucus. This immune response is able to stop the IB virus from attaching to the cell. Through binding to the spikes, the local antibodies generated by the vaccination prevent infection. Unfortunately, there are many different variants of IBV with differences in their surface proteins or spikes. Highly specific antibodies are able to recognize one type, but usually not others. This is why, despite vaccination, a different type of IBV will be able to infect the birds, causing infectious bronchitis. IBVs can occur in many so-called serotypes that differ in the structure of their surface proteins or spikes. Some serotypes of the IB virus show marked differences, but others possess a large degree of similarity, allowing them to be classified in groups. Certain serotypes are widely used as vaccines because they are able to induce immunity against more than one serotype. From a practical point of view, it is therefore more relevant to talk about protector types rather than serotypes. The Massachusetts MA5 vaccine represents one such protector type. Vaccination with it leads to broad protection, preventing a wide variety of IB viruses from infecting the birds. This phenomenon is known as cross-protection. But due to the wide variety of serotypes of the IB virus, vaccination with one protector type alone may not be enough to immunize the birds. This can result in IBV problems even in vaccinated flocks. For a broad level of protection, the use of two vaccines representing different serotypes is recommended. This is referred to as the protector type approach, which classifies viruses according to the way vaccinated chickens react to them. The MSD animal health vaccines Nobilis IB-MA5 and Nobilis IB-491 provide broad protection against infectious bronchitis in the flocks. The vaccines are easy to use and can be administered by spray, eye drop, or in the drinking water. Using the protector type approach, the following vaccination protocol has proven most effective. Nobilis IB-MA5 on day 1 and Nobilis IB-491 on day 14. This will protect the birds against most IBVs during the first weeks of their lives. To safeguard egg production of layers and breeders in the long term, repeat vaccinations prior to the onset of the laying period are necessary. These should also include the use of an inactivated multivalent vaccine. The efficacy of these two vaccines has been widely proven using the ciliostasis test, a tool to measure the impact of a virus on the cilia of the trachea. For this test, samples from different areas of the trachea are taken and ciliary activity is measured. The ciliostasis test also evaluates the level of protection after vaccination. Values above 50% represent good ciliary activity and therefore good protection. Values below indicate insufficient protection. The use of the two vaccines achieves far better results than using them individually. MSD Animal Health Nobilis IB vaccines, based on the innovative concept of protector types, provide a very good level of immunity and protection against many serotypes of the infectious bronchitis virus. Mass vaccination is uncomplicated and helps the poultry industry avoid the considerable financial losses associated with the disease. MSD Animal Health Vaccines. Broad protection against infectious bronchitis.